Maybe we'll start for the exam. I don't know. How's everybody doing?
Good.
Everybody's smiling. This is good. Somebody handed this to me at the beginning, said it ended up in their backpack accidentally. It's somebody's biochem notebook. If it is yours, it's here to claim. So come and claim it. If you have not gotten a note card yet, please remember you're required to have a note card that you get from me for the exam. And I will have those. I have those in my office. If you want to come by, I will give you one. But if you don't have a note card for the exam that you got from me, you will be penalized points. So make sure you've got a note card from me. It doesn't have to be filled out. It does have to have your name on it, though. And that's something to keep in mind as you're filling out your note cards. As I said in an email I sent to everybody, the note cards must be 100% handwritten. Okay? No printed figures, no pasted on things, 100% your own handwriting that's on there. And it needs to have your name on there so we can obviously know that you turned one in. Julie?
You said you wanted them full. How full do you mean? Like, you want every single space filled?
The question is, do I want them full? It's up to you. You can leave them blank if you want. I'm sorry?
You said the other day that you wanted them filled out. So
No. You can have a blank card if you wish. What you put on it is up to you. Some people don't want to use a note card, and that's fine, but you still have to get one and turn it in. Okay? Now, I make you turn that in, and I don't give those back, and the reason I do that is so that you will not pass it on to your roommate next year. There's a benefit of you filling it out, and there's going to be a benefit of your roommate filling it out next year. So if you pass your card on to your roommate's not going to have that benefit. So that's why I collect those cards, and I don't return them. So if you want what's on there, photocopy them or whatever, that's fine. But obviously, they'll have to fill out their own note card next year. Okay? Okay, I have almost finished regrading the exams, and I'll be putting them in the BB office probably about mid-afternoon today. You can pick those up from the BB office if you want to look at those. Have those as a guide for studying. I haven't yet announced a review session. I will announce that probably in an email and online, so you can see where and when that'll be. That will almost certainly be in ALS 4001, as the other two have been. I just don't have a time yet scheduled for that. Yes, ma'am? Can the note cards be written in different colored ink, highlighted, both sides, etc.? All those, answer is yes. The one thing I won't let you do, and this was an interesting thing that somebody did a few years ago, was they wrote it in different inks, and they did one card with, they brought in glasses, so they had a red and they had a green. And so they could literally read twice as much information. And I said, perhaps that's a little over the top. But it worked. I mean, it worked. I look at this thing, it's like, holy moly. I mean, I can see in the red lens, I can see one set of things, and the green lens, I can see something else. So,
I think that was great.
The creativity, I'll tell you that the creativity that I see on these is organization. People are really amazing at organizing these things, and how small people can write. My God, that's incredible. Some of it's so small it's hard to read. And no magnifying glasses either. That's the other question, right? Bifocals are kind of hard to... I have bifocals, but you can't tell, so they're kind of hard to... If you have bifocals, okay. So, all right. Other questions? Yeah?
How is the test, is it longer?
Oh, a good question. So the exam will be about 150 points. Okay? So it's about 1.5 times as long, but you've got twice as long to take it. So time should not be a factor. We'll give you the full hour and 50 minutes, which is 110 minutes. We'll give you the full hour and 50 minutes to do it. The exam... Well, that's not good. That means I get to yell. Okay? So, what's that? No, it's not doing anything, so I don't know. That's doing something, though. All right, so it's about an hour and 50 minutes that you get. The exam is in here on Thursday at 6 p.m. Thursday, December 9th at 6 p.m. Mark it into your brain. Okay, so it's been a really good term. I have to say I've really enjoyed meeting many of you. So a few of you I knew ahead of time, but I've enjoyed meeting and getting to know many of you, and it's been a very nice term for me. I've really enjoyed that, and I've enjoyed those interactions. And I know for many of you, we'll have those interactions again next term, and so I actually look forward to that as well. I hope you do. You never know, but I really have enjoyed that, and I thank you for all that you bring to the class. Well, we have to finish things up today, and as I said, I have a little surprise for you. And the surprise is even a surprise for me, I have to say, and we'll see why that plays in as we get a little further along. So, last time I talked about this figure, and this is about the place where I stopped. And the place where I stopped was the regulation of the synthesis of glycogen. Now, we talked about reciprocal regulation. And reciprocal regulation, I will remind you, has opposite effects on catabolic and anabolic pathways. So this is a really nice depiction of a reciprocal regulation that's going on inside of a cell. This could be, let's say for the moment, a liver cell. Okay? Now, we look at this, and what we see is that this cascade, which starts with either epinephrine or glucagon, results in the activation of the enzyme that breaks down glycogen at the same time as it results in the inactivation of the enzyme that synthesizes glycogen. Well, this makes very good sense. We don't want synthesis and breakdown going at the same time because we're trying to avoid a futile cycle, right? We don't want to have a futile cycle. It's just going to burn energy and waste energy and not do anything more than that. What we don't see on this figure is how this pathway is reversed. Okay? So we see how to break down glycogen and we see how to turn off glycogen synthesis, but we don't see how to turn on glycogen synthesis and turn off glycogen breakdown. Well, as you might surmise pretty quickly looking at this figure, the things that turned things on here were phosphates. The kinases were very active. The things that we don't see are what's turning that off. So you might imagine that removing those phosphates will have the opposite effect. And in fact, you would be exactly correct. By the way, as I'm saying this, I'm thinking of one very cool thing. Several of you sent me the news article yesterday of the microorganism that was found in Mono Lake. If you haven't heard this, I'll tell you here. It's a phenomenal thing. They have found that this organism can grow in the absence of phosphate. Phosphate is thought to be was thought to be one of the essential molecules of life. And it turns out that you can have this completely in the absence of phosphate. It can grow. And what it's growing in is arsenate. And on the periodic table, arsenate has chemical properties similar to phosphate. This thing puts arsenate into DNA and building DNA. An absolutely remarkable thing. Now, relevant to kinases, what do kinases put onto proteins? Phosphate. What do you suppose this organism puts onto proteins? Probably arsenate. And where does the arsenate come from? There's a good exam question. How would the arsenate get onto a protein? What would have to be the precursor for the arsenate to make it onto the protein? Okay, I'll give you a hint. These cells aren't going to have any ATP. Why? No P. What are they going to have in place? They're going to have ATA. Yeah. Adenosine triarsenate. Isn't that wild? Okay, you think about this. And they have evidence. This is very much in the early stages, but they have evidence that in fact both the DNA is full of arsenate, no surprise, and some of the proteins have arsenate on them as well, which would indicate that what we just said, putting arsenates onto things, is in fact probably what they're doing. Okay, so, all right, but we're not going to deal with the biochemistry of those cells right now. But they're kind of a wild thing. All right, so when we look at this, we see, as I said, we turn on breakdown, we turn off synthesis. Let's now consider how we would turn off breakdown and turn on synthesis, okay? That's actually shown, nope, wrong one, sorry. I do this all the time. That's the wrong one, too. What did I do? Here we go. No, that's not what I want either. I look at, believe it or not, I do look at these before I come to class. Here's what I want. All right. Now, this is a dumb figure. I don't like this figure because I think it's confusing when you have glycogen synthesis required, blah, blah, blah. Okay? So I like to think of this figure in very simple terms. Coming from the top down, moving from the top direction down, it's describing to us what happens when we treat, for example, a liver cell with glucagon or epinephrine. Okay? We treat a liver cell with glucagon or epinephrine. We see that protein kinase A is activated. You've known that before. You see that phosphorylase kinase becomes activated by protein kinase A. You see phosphorylase kinase activates phosphorylase A, etc. Okay? We see that protein kinase A inactivates glycogen synthase. We see the red as the inactive forms. The blue is the active forms. Okay? Now, if we start at the bottom, what we're seeing is what happens when cells interact with insulin. Now, insulin is a hormone that, as I've talked about before, counteracts the effects of epinephrine or glucagon. All right? It stimulates exactly the opposite sorts of things, and it makes good sense. Because why do cells make insulin? They make insulin because they're trying to reduce the blood concentrations of glucose, because glucose is a poison, and they're telling these cells, take up glucose. Well, you just told cells to take up a poison. Right? So how do cells deal with this? Well, they don't leave a lot of free glucose laying around. They decide to do something with it, and they stimulate the synthesis of glycogen, which uses glucose. Glycogen is not poisonous. So when insulin attaches to the insulin receptor, okay, in addition to that glut protein that you saw before that got moved to the receptor surface that brought the glucose in, this is showing what happens, how to deal with that glucose once it's inside the cell. Insulin treatment of cells activates an enzyme known as protein phosphatase 1. I also call it phosphoproteinphosphatase 1, so sort of flipping those two, whichever, either of those is acceptable. Okay? This enzyme is a phosphatase. A phosphatase is the opposite of a kinase. A phosphatase removes a phosphate. And this is very simple, because what this guy does is it removes phosphates off of all of those things that we saw before. It removes the phosphate off of synthase. That has the effect of activating the synthase. It removes the phosphate off of glycogen phosphorylase kinase. It removes the phosphate off of glycogen phosphorylase, which leaves it inactive. It removes the phosphate off of phosphorylase kinase to leave it inactive. And we're all set. Protein kinase A isn't being stimulated for a couple of reasons. One, of course, is epinephrine isn't being produced. And the other is, what was the thing related to caffeine? Phosphodiesterase is being active, and it's breaking down the cyclic AMP, so protein kinase A isn't active. So we've reversed the entire process simply by treating the cell with insulin. Insulin has a counteracting effect to that of epinephrine or glucagon. Everybody with me on that? Questions on this? No questions today at all? Let's see if I have a microphone or not. I have to yell so much. Oh, I just turned it off. Duh. Okay, now hopefully I don't have to yell. All right, so that's what's happening with this pathway. So it's really phenomenal what's going on with this reciprocal regulation. Now, what I haven't shown you is, well, there's always something else, right? And the something else that's always there, in this case, is, well, what happens with, how do we control protein phosphatase 1? Okay? That's what I'm going to talk about next. Turns out protein phosphatase 1 is regulated by a molecule called PI1. Okay? It's also called in this figure inhibitor. So we're going to call it, I guess we'll just call it inhibitor. How about that? Keep it simple. So when we talk about inhibitor, it's a specific thing that's specifically inhibiting PP1. PP1 is protein phosphatase 1. This is that abbreviation. PP1 stands for this. Okay? If you want to call inhibitor something more specific, you can call it PI1. In fact, when you look in the highlights, I'll write that as PI1. But inhibitor, I'll accept that as well here. All right. Now, this shows us something very interesting. Okay? Here is a protein called G. It's not a G protein. It's just called G. And it has the M due to the fact that they're looking, in this case, in a muscle cell. So they've got G there. And G associates with phosphoprotein phosphatase 1 or protein phosphatase 1. Same thing. All right? When these two are associated, as you see here, protein phosphatase 1 is active, meaning that insulin is bound to the cell. So whenever we have insulin bound to the cell, we see this situation shown on the left. Well, how do we turn off protein phosphatase 1? Well, you said that insulin was counteracted by epinephrine and glucagon, right? So if glucagon and epinephrine bind, don't look at the figure, I want you to think. All right? Glucagon and epinephrine bind, what do they stimulate in the cell in general? Okay, general. Kinase action. They stimulate phosphorylation of proteins, and they do that by ultimately stimulating, via cyclic AMP, protein kinase A, right? All right? So they stimulate phosphorylation by stimulating protein kinase A to be active. Now, look what happens here. Here's protein kinase A. In addition to all the other things you've seen it phosphorylate, look what else it phosphorylates. It phosphorylates the G. So I'm calling this G and not G protein. Okay? So it phosphorylates G. When it does, look what it does. G loses its affinity for the protein phosphatase 1, which leaves it in a much less active state. In addition, protein kinase A phosphorylates the inhibitor, which allows it to bind to PP1. The combination of these two things leave the protein phosphatase 1 completely inhibited. This means that epinephrine stimulation completely inactivates the protein phosphatase 1. Okay? Now, I'm not going to show you how we go backwards from here, but suffice it to say that insulin stimulation will reverse this process. Insulin stimulation will reverse this process. When we reverse the process, protein phosphatase 1 becomes active, and everybody's happy. Okay? Questions about this? Oh, I heard my name somewhere. Yeah. Yeah. So, when we're on the left side of this figure, insulin is bound to the surface of the cell. So we're looking at insulin stimulation over here. We're looking, once we get over to here, we're looking at epinephrine stimulation here. So they have opposite effects. Insulin stimulation leaves this guy active. Epinephrine stimulation leaves this guy inactive. Yes, ma'am?
And you said pKa phosphorylates the G.
pKa phosphorylates two things. It phosphorylates the G, which is why it lets go of the PP1. It also phosphorylates the inhibitor, which causes it to bind to PP1. Okay? Yeah, I know there's a lot of complexity here. But if you look at this in reciprocal terms, everything adds up. Everything is consistent. Okay? Everything is consistent here. We're having opposite effects if we do epinephrine versus doing insulin. Okay. And you'll notice, I meant to also mention, your book talks about doing exercise or fasting. It's not even clear what the hell they're talking about, left, right, etc. Don't worry about exercise or fasting. All right? If I told you that you ate a meal, what would you tell me was being produced? Insulin. If I told you you was running away from a grizzly bear, what would you tell me was being produced? Epinephrine. Okay? That makes a lot more sense than, which part of the figure am I exercising or eating or drinking or what? It doesn't make any sense, right? Your book is dumb in a couple ways. That's one of them. Okay. This is good. Okay, so I need to tell you something that's very surprising that was discovered about glycogen breakdown. Oh my God, there's another thing about glycogen breakdown? Yeah, there is. Okay? So when we look at glycogen breakdown, there's something that people discovered a few years ago that was very surprising to them. Now, I've got to describe the experiment that they did. What you see are the results of it on the screen. They took and they isolated from liver. They had glycogen phosphorylase in the A form, meaning it had phosphate on there, meaning it was what we think of as the more active form. They also had in this tube glycogen synthase in the B form, meaning it also had phosphate on there. Okay? So they got glycogen phosphorylase with phosphate on it. They've got glycogen synthase with phosphate on it. And they're sitting here in this tube. And they want to measure or what happens to these enzymes. Okay? They add glucose, and something very surprising happened. Okay? Now you would think, well, we're going to add glucose. Nothing really should happen because neither of these enzymes uses glucose directly. Remember, they use glucose 1-phosphate or UDP glucose to synthesize, or they produce glucose 1-phosphate and they break it down. But all they did was they added glucose to this, and when they added glucose, something very surprising happened. The surprising thing was, and again, these were the only two things were in the tube: phosphorylase A and synthase B. At least so they thought. When they did that, they discovered that phosphorylase A quickly converted to the B form, and glycogen synthase B quickly converted to the A form. Now, what has to happen to those two proteins in order for that to happen? The phosphates have got to come off. Well, what takes phosphates off? Phosphatases, right? Well, all of a sudden they discovered that the phosphatase was hitching a ride. They didn't know that before. The phosphatase was hitching a ride, and in fact what it was doing is shown right here. Here we are in the liver. It turns out that the phosphatase is linked to a liver G, that's the L right there. It's linked to there, and it's normally carried, this is amazing, by the glycogen phosphorylase A. It's carried by the glycogen phosphorylase A. When it's being carried in this form, the phosphorylase A is blocking the PP1 from being active. So even though it's associated with the G, this phosphorylase A is blocking the G from being active. Okay? When this guy then encounters glucose, look what happens. Glycogen phosphorylase A converts from the R state to the T state. Have you seen this before? You did see this before, because this was one of the allosteric regulators of glycogen phosphorylase A. When glucose was present, what did I say happened to it? It goes to the T state. It's less active, because when cells have plenty of glucose, they don't want to be breaking down more glycogen. The reason that this happens is partly because these guys are attached. It becomes detached, and now the phosphoprotein phosphatase, the PP1, is released, and now it's active. Guess what it does? It starts dephosphorylating everything. Now that's really cool. It means that this guy right here on the left is basically a loaded gun. And that loaded gun, okay, is ready to spring into action as soon as glucose concentrations start to accumulate. This is a hair trigger that's on this system that shuts down glycogen breakdown the instant glucose starts to accumulate. This makes sense. Enzymes are very efficient. They can break things down very quickly. If you break down a whole bunch more glycogen than you need, you're going to have to turn around and resynthesize it again. That's going to waste energy. This allows that system to turn off very quickly, and it's turning off the glycogen phosphorylase. What's it doing to the synthase? It's activating it. And what's the synthase going to do with that extra glucose that's floating around the cell? Ultimately, it's going to make it into glycogen. Yes, sir? Free glucose, that's right. Completely free glucose. So synthase can't use completely free glucose. That's why I say it's going to ultimately do that, because the free glucose is going to get converted into glucose 6-phosphate, to glucose 1-phosphate, and then into UDP glucose if it's going to make it. Right? Yes?
So originally they thought they just had phosphorylase and synthase, but what it was is the phosphorylase had
The phosphorylase had G with the phosphatase, and they didn't know it. That's right. Yeah.
the G and the PP1.
Right. Yeah. Kind of cool, huh? Everybody says, what's the surprise, right? Tired of this crap. Yes, question, Shannon.
What is G again?
So G is just the name of a protein. It's not a G protein, but it's just what they call this protein that's found in liver.
It's not an enzyme or anything? It's just
It's not an enzyme. It's a binding protein that hangs on to PP1. So when it's hanging on to PP1 by itself, it's active. In this case, the PP1 is blocked, so it can't work. Okay? And in this state over here, it's very active. And so it will take phosphates off of things. Did you hear my question? Okay. All right. Any other questions there? Well, we're not quite at the surprise yet, because, as I said, the surprise is a bit of a surprise for me at this point. So I've got to stall. All right. That leads me to my last thing I'm going to talk about, and there's my signal back in the back about where I am. So, okay, I don't know what my signal is yet. So, okay. Santa Claus is coming to town. All right. And I've been very bad this year. I can tell you that. So, all right. So, what I'm going to show you here, you're not responsible for, but I think it's interesting. Are we a, or are we a? Oh, okay. Well, maybe we are. Maybe we have a good surprise, and that's good. Okay. So, glycogen is, as you might imagine, a pretty important thing for an organism. Everybody's looking back now instead of looking at me. We don't have to know this, right? So, trust me, it's not going to go away, okay? And no, it's not Santa Claus, okay? Although, well, okay. Glycogen is a very important compound for an organism, because in an animal, I said it was a storage form of glucose, and it allowed the organism to be able to release glucose very quickly. What's very surprising is that there are some significant mutations that can happen to some of the enzymes that I've described to you, and the organism can still be alive. Okay? Now, this one here, to me, is the most interesting one. Okay? It's called McArdle's disease. And McArdle's disease occurs when a person is lacking glycogen phosphorylase in their muscle. Now, I would think this would kill a person, right? If I'm just guessing, I'm going to say this is a pretty important thing. I've got to be able to release glycogen, glucose from my glycogen so that I can run and escape, et cetera, et cetera. But it turns out the people who have McArdle's disease live. And the main thing they have is when they exercise, they feel some pain of exercising, especially at first. Now, based on what you know about biochemistry, I'll give you the question here so you won't have it on your exam. Based on what you know about biochemistry, how is this person able to stay alive and exercise? There's one pathway that you've heard about, or one, not pathway, but one sort of system that you've heard about that allows this person to be alive. Yes, what is it? The Cori cycle. This is an excellent example of what the Cori cycle actually allows a person to keep alive. Let's think about this. I've got McArdle's disease. My muscles don't have phosphorylase so they can break down glycogen. So they've got glycogen sitting there and they can't do anything with it. They take off on a run. They very quickly exhaust their glucose. In the normal person, they would start breaking glycogen down very quickly. This person can't do it. That's why they feel the pain. The muscle cells are straining. The muscle cells are hurt. Okay? The brain is already kicked in. It's already started dumping epinephrine, and it's gone to the liver saying, hey, this guy's going to need, or girl's going to need, some glucose, and the liver starts producing glucose. This is the Cori cycle. Glucose gets dumped in the bloodstream. It goes to the muscle cells. The muscle cells grab the glucose, and now they've got energy to do their thing. It's an absolutely remarkable thing. Now, obviously, this person has to have a phosphorylase that functions in their liver. A phosphorylase that doesn't function in the liver is probably going to be very, very deadly. Okay, let's see. So I think if I could get my little Santa Claus back in the back to give me a signal. Santa. Are the elves ready? Okay, so you can put your pens away. In fact, if you don't want to hear some bad singing, you can leave. Okay. I have a little surprise for you, and every term, as you know, I like to sing songs. Every term, at the end of the term, I always write a couple new songs for my classes, and this term is no exception. So I've got a couple of new songs for you, and not only that, I have a surprise to go with those songs. So I'm going to show you the surprise, and you can... It comes with history. How many of you guys didn't think this was a history class, but in fact, there's an element of history in everything that we talk about. This element is the history of music. Okay? And the history of music, if we look at music, it really comes down to a set of Bs. All right? For example, okay, all music started with the Beatles. All right? That's given, right? I mean, it started with the Beatles. And in fact, more importantly, I think that the Beatles should have been like that, okay? Well, the Beatles were a big B group. They led to another group called, in fact, in this case, the Bee Gees, which probably you guys, none of you guys even remember them, which is probably just as well. And this gave rise to I mean, everybody loves these guys, right? Okay? And, well, then these guys gave rise to something even worse, which was, yeah. So, well, in keeping with that tradition, we've decided to introduce the Biochemical Choir. And I'd like to introduce them right now. So if the Biochemical Choir would come forwards, we had several illnesses, so I wasn't sure if we were going to have a choir or not, but we do. Choir, that's your cue. So I want to thank the choir for coming and singing. We had about a dozen people, but we had several people who got sick, and so that's why I didn't know until the very last minute if we were going to have a choir. We have a choir, and I'm very happy to see everybody here. So this is their Oops, We're Going to Do It Again tour. And if you guys want to come maybe right over here.
Can you turn the damn camera off?
All right. Let us thank, first of all, Aresta, and we have Kalia, we have Minhauser, we have Indira, and we have T, and we have Phil, Dr. McFadden. So Dr. McFadden teaches the BB 490 series, and we're happy to have everybody here. So thank you guys for bailing me out.
Can you turn that off so that you're not blinking?
I can't, unfortunately, because they've got to see what we're going to be doing. So maybe if you squat. Sorry. Okay, so to do the first song, we got to get in the right mood, okay? The right mood. So there's an old-time song back in about the 30s that was very popular, and we have to get started thinking about this. It was back in the war times, back in the Depression, okay? And people were very unhappy. And this song goes, happy days are here again, the skies above. Join me. Happy days are here again, the skies above are clear again. Let us sing a song of cheer again. Happy days are here again. Come on, I gotta hear you. Happy days are here again, the skies above are clear again. Let us sing a song of cheer again. Happy days are here again. Now continue. Crappy days are here again, the skies above are clear again, and the sun has disappeared again. Crappy days are here again. Rain is falling from the sky. I wish I knew the reason why. Guess I'll have to wait until July for the weather to be dry. I do not mean to harangue, since rain provides yin and yang. Because the flowers, everyone, love moisture followed by the sun. Let's stay happy till the rain is done in Corvallis, Oregon. All right. As you guys know, I get out of breath when I do these, so. Okay. We are next ready for something that relates to something we heard earlier this term. It's a song that we've done before, and this one I think you're going to all relate to. Everybody know this song? Raindrops keep falling on my head.
Can you pick a key we can sing in?
You guys started.
I don't sing it.
Brain farts keep just happening in my head. I think it might be due to something Kevin said. Biochemistry gets brain farts a-popping in my head, and they're popping. So I just wiped out the teardrops from my eyes and told my brain it had to do some mental exercise, burn some ATP, so brain farts can stop inside my head. They'll be stopping, 'cause there's one thing I've learned. When energy increases, it sure pleases my mental state. I'm doing great as tension eases. Now brain farts don't happen in my head, so I'm sure the final will be easier instead. Cyclic AMP stops brain farts from popping in my head. They're not popping thanks to caffeine. Nothing's worrying me. All right. Okay, so those are the two songs we've sung in class, or at least I've sung to other classes before. Now I have two new songs just for you guys. The first one, how many people actually remember the Beatles? Nobody here remembers them. But you've heard the music, right? Well, I'm old enough that I remember them when they were running around. And they had a song that I really loved. It's called I Will. Does anybody know this song? Who knows how long I love you? I know I love you still. Anybody know that song? Oh, boy. This is going to hurt, especially since our choir hasn't been practicing this song either. So this one is about muscles. It's called the Muscle Song, and it's tuned to the tune of I Will. Okay? So I'm afraid I'm going to have to probably start this one. So for running and for jumping, you need some energy. Chemically, the body stores it in the form of ATP. If backup should be needed, reserves are there in weight. Muscles brimming with supplies of tiny creatine phosphate. Ready whenever cells are ever needing to exercise. Steady as ever when whatever energy needs arise. The action is exacting for leaping in the air. Myofibrils all contracted, using energy extracted from reactions that react in me, using ATP. You see? Okay. Okay. I promise this next one.
You should make everyone sing the last one together.
Everybody's got to sing the last one together. So we're gonna, if we want to go out on a high note, everybody's got to sing this one, okay? This one's a tune everybody knows. It's Winter Wonderland, right? Da 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 da. Right? Everybody knows it's Christmas, right? Everybody's ready for this? Okay, this guy is called. It's the last one, so we got to be loud, okay? The louder you are, the easier that exam is going to be. I'm writing that exam. Okay? Who wants to have an extra credit question on that exam? Okay. If I don't hear you loud enough, there's not going to be one. So we want to belt this one out, okay? This guy is called BB Wonderland. Are you ready? Okay. Go. Milam Hall, it's 12:30, and Ahern's getting wordy. He walks to and fro while not talking slow, giving it to BB450. I was happy when the term got started. Lecture notes and videos galore. MP3s got added to my iPod. But recitations sometimes were a bore, and exams bit me roughly when the curve turned out ugly. I don't think it's so, my scores are too low, sliding by BB450. Finally, there's an examination on December 9th at 6 p.m. I'll have my card packed with information, so I don't have to memorize it then, and I'll feel like a smarty with my jam-packed no cardi. Just one more to go, and then ho ho ho, I'll be done with BB450. That's it. Thank you guys, it's been a great term. Thanks to the Biochemical Choir for bailing me out here. Thank you guys very much. Woo. All right. See you guys on December 9th at 6 p.m.
Are you going to release this?
I will, as I said, I'll announce it. Yeah.
if we need to get no cards still, do
Yes,
just
you go
to
see
your
me.
office?
Go see me. Uh-huh.
Are you going there now?
I'm going there now.
Okay.
How you doing? Is that yours, Daryl?
Yeah.
Oh, okay.
that's the other class.
Somebody turned it in, so good.
Kevin, do you have any note cards with you? Or are they?
No, they're in my office. Yeah.
Okay.
Did you hand out no cards?
They're in my office. You'll have to come to my office again. Okay?
Thank you.
Uh-huh.
You gotta do the tunes we know.
Yeah, well, I'm an old guy. What can I say, you know?
What are you gonna have the slides posted for last time and today?
Last time is posted last night, and
Okay.
today's will be posted as soon as I can get them up there.
Okay, thank you.
Okay?
May I get a note card from your office when you're in your office?
Yeah, sure, sure.
Will you be there in about an hour? Like, this afternoon?
I'll be there. Should be, yeah.
Okay. Okay. Just have a class.
Yeah, yeah. Hi.
How are you?
I'm fine. How are you?
I'm okay. So I had a question about the arsenic.
Okay.
If that's
Yeah, sure.
okay.
Pretty wild, isn't it? So arsenic does several things. One, it's very reactive, so it can combine with proteins and cause some major problem just from the chemical reaction with the proteins. But probably the more important component, we'll talk about it next term actually, the more important component of toxicity of arsenic relates to the fact that it inhibits an enzyme that's necessary for the breakdown of pyruvate and another molecule in the citric acid cycle.
Okay.
And so as a result, you get no energy. So
My understanding was that it fell off into. Okay, that's